Okay, so we're going to discuss about the HIV. So, HIV, the human immunodeficiency virus, is causative agent for AIDS or your acquired immunodeficiency virus. HIV virus has been identified by Gallo, Montagnier, and we have also Levy of the US. Okay, so again, we have here the HIV has been discovered by Montagnier, Gallo, and Levy. So other name for your HIV virus, we have your human T-cell lymphotrophic virus 3. The human T-cell lymphotrophic virus 1 and 2 are those are your oncoviruses. Pag sinabing oncovirus, means say they are causing here cancer, they're cancer producing. But the human T-cell lymphotrophic virus 3 is responsible here for the HIV. And also called here your lymphadenopathy associated virus. And we have also here your AIDS-related complex. Basically, we have here two HIV. We have here your HIV-1 and HIV-2. HIV-1 is made up of 9 subgroups, divided into your M group, and we have the N and the O subgroups. And we have here the HIV-2, on the other hand, made up only of 2 subtypes, subgroups, Group A and Group B. Okay, the virus belongs to the family of retrovirus and belong to the subfamily of the Lenteviridae. So basically, the virus is spherical in shape, and the nucleotide of the virus is RNA. This is a single-stranded RNA. And like for example, this one is our virus. We have here, so at the middle portion, we have the single-stranded RNA, and it's being enclosed by a nucleocapsid. Plus, meron pa rin siya ditong outer structure in the form of the envelope. This is an envelope na virus. And the envelope here is actually came from the cell membrane of the infected na cell. Okay, now we have here the different genes utilized by the virus in order for it to become its structures. So divided here into your envelope gene. The envelope gene give rise to the GP or the glycoprotein. Ang pinaka-precursor na glycoprotein, we have here GP160. And the GP160 eventually give rise to GP120. The GP120 is a knob-like structure. This is the outer globular structure which is being utilized by the virus in order for that to attach to the CD4 positive cell as its main target na cell. And we have also here the transmembrane protein. We have here your GP41. So for example, this one is your virus. So this is yung ating GP120. This is your transmembrane protein. This is your GP41. The next gene of the virus here code for its production of its protein structure. Okay, that's basically the GAG gene. And we have your pinaka-precursor niya, P55, which eventually give rise to the following proteins, P6, P9, P17, P24. And the P24 ay core protein niya. Ama ito ay galing sa precursor substance na P55. And another gene, we have here the Pol gene, polymerase, essentially responsible here for the production of the enzymes of the virus. So this includes here your P56, P66, P57. Okay, that give rise here for the production is reverse transcriptase. Reverse transcriptase is the enzyme utilized by the virus in order for that then to reverse transcribe its RNA para maging DNA. That would enable it then to insert to the host DNA. Then P31 give rise here to your another enzyme, integrase. Again, it's utilized by the virus after na ma-transform ang kanyang RNA to become DNA. That DNA of the virus dapat insert niya sa host DNA with this enzyme. And lastly, we have here the P10, this is for the protease. Okay, another one, we have here the different accessory proteins of your HIV. So, kindly copy na lang. Okay, next, we have the difference between your HIV-1 and the HIV-2 in terms of their structure. So, the globular protein knob-like structure for the HIV-1 is GP160, 120. Sa kanya HIV-2, we have here your GP140. So, we have also here the transmembrane glycoprotein. For the HIV-1, it's your GP41. Sa ating HIV-2, that's your GP34. And it just also differ with their core proteins. P24 for HIV-1, HIV-2 is your P26. So notice na ang kanilang pinagkaiba ng HIV-1 sa HIV-2 natin will have in terms of their GAG gene and their envelope gene. So their Pol gene, polymerase gene, or their enzymes, they almost have the same on that in terms of that structure. Okay, then we have here the different mode of transmission of the HIV. So you get infected by number one, sexual. Could also be infected here by parenteral. Parenteral is just direct inoculation of the virus. It could either be because of the accidental needle stick puncture, IV drug user, or even blood transfusion. The third one, perinatal, that's vertical transmission. So mean to say the mother is infected, and eventually the baby could also be infected with that because they are trying to share here their blood. Then we have here the viral replication. So the virus here try to infect our cells here. Basically, yung pinaka-target nito is your CD4 positive cell. Your CD4 positive cell here could be your T helper, could also be your monocytes, macrophages, and dendritic cells that are also CD4 positive. So if your virus try to infect the T helper cell, you call this one as the T-trophic na strain, or you call this one as the X4 na strain of virus. Those HIV virus will try to infect your monocytes, macrophages, aside from your T helper, you call that one as your M strain, or you call that one as your M-tropic or your R5 strains. So again, ginagamit ang virus natin ang kanyang GP120 in order for that to bind to the CD4 positive cell. What mediates your virus here to attach, okay, basically to your CD4 positive cells here is by your chemokines. Example of chemokines, so we have your CXCR4. So if that one, ang virus natin nag-attach sa ating T helper. On the other hand, it uses the CCR5 na chemokines if your virus here try to attach the macrophage. Again, the chemokine helps here in order for your virus to stick to the surface of your CD4 positive cells. And after that one, after the adherence, the virus here undergo endocytosis. So, papasok na siya sa loob ng cytoplasm ng mga CD4 positive cells natin. Once the virus is already inside the CD4 positive cell, it tries to uncoat. Encoding here, it tries to release its viral RNA. Once it tries to uncoat, the viral RNA will be transcribed to your DNA by the reverse transcriptase na enzyme na ginagamit ng virus. So, pag naging DNA na kanyang viral RNA, next here, it need to attach or to integrate that one to the host DNA by using as another enzyme integrase. So, pag inattach na niya ang kanyang DNA sa ating host DNA by the enzyme integrase, what's going to happen next here, your host DNA would undergo replication, transcription, and translation. Since naka-integrate na ang viral RNA or the naging DNA na siya, still as your host DNA here try to undergo that processes, replication, transcription, and translation, still also, alongside with that, your viral DNA also undergo that process. And therefore, ang pinaka-product nito ay mga viral products. So, ang gagawin ni virus, pag nakagawa na siya ng mga byproducts niya, it try to assemble that one to become a new virion or new virus. So, pag magkaroon na siya ng new virion, time will come that your infected cell will going to die, then the virus here will going to come out of that infected cell, and the infected cell here will die. And along with that, as your virus try to buzz off from your infected cell, it try to carry the cell membrane of the infected cell. Kinukuha ng cell membrane para gawin niya ang kanyang envelope. And therefore, your virus here is an envelope na virus. And then again, that envelope came from your cell membrane of your infected na cell. And after that one infected cell die, and the new virion will now again go to the next CD4 positive cell and try to infect it again. And the cycle goes on and on. Okay, then we have here the immune response of the patient here once you have the viral replication process. So number one, the patient would have here, with the initial replication process, would have here the viral burst, followed by slowing down of the viral replication. But because of the product of the viral replication, the patient will be positive for the P24. So this is the first serological markers that will be present if the patient would have infection with the HIV. But then again, a problem with the P24, the first two appear, but that marker here will not persist for longer period of time. So madali din siya nawawala. And therefore, we could not really rely on that marker for us then to diagnose someone na may HIV. The next thing here, because of the viral replication, then eventually the viral protein will be attached to your MSC class 1 and that will be presented to your B lymphocytes. And that result here to the activation of your B lymphocytes that result to your antibody production, activating your humoral mediated immunity. Okay, the first antibody to be produced directed towards your GAG gene P24, but then again, the antibody is short-lived, nawawala siya, it disappears faster or immediately. Then next, the body try to produce an antibody directed towards your envelope genes, yung mga glycoproteins natin. GP120, GP41. So, pinaka-ideal marker for that is your GP41 because that marker would eventually try to persist for a longer period of time if you have your HIV infection. Then you also try to produce antibody directed towards your polymerase gene, and we have also your accessory proteins. The next one will activate also your NK. It will activate your NK through the antibody-dependent cell-mediated immunity. And then another one, it will also try to activate our cytotoxic cell, T cell, or CD8 positive. That's your cell-mediated immunity. But then again, although our body is responding to the infection, but all of that immune response here will not provide us with the protection because it could not really remove or kill the virus completely. So our markers here, our antibody produced here are only for serological markers for us then to diagnose or to monitor the course of the entire disease process, but not providing us with the protection.